This is finding Frankenflora using PCR to rapidly detect hybrid proteas. This project is important for South African biodiversity because natural famous hybrids are difficult to identify by morphology after the first generation of parental fire. Hybridization could lead to a loss of genetic diversity. With genetics, we send lots of samples away for sequencing, which is expensive. So why am I using the word Frankenflora? Well, in 2001 and 2005, Dr. Tony Ribello came up with this idea of the Frankenflora. Frankenflora are dominant hybrid plant game changers that result in the loss of populations and potentially threaten highly localized species. In the Proteaceae groups, there are 30 that are vulnerable to hybridization. This is an informal assessment. Publications on hybridization in the wild of Protea species since 2005 have been 1. Reopening the Case of Frankenflora by Timothy Macqueen and Alistair Potts. Dr. Tony Ribello referred to them as Frankenflora because he wanted to show the monstrosity of these plants like the Frankenstein's monster. So originally we came up with this idea of the hybrid identification toolbox, which would use plant morphology such as the scent and the size of the leaves, sending a few individuals for sequencing, using the single nucleotide polymorphisms, using PCR of the internal transcript spacer from the nuclear ribosomal cistron, and also using screening by high resolution melt analysis or HRM. There are 2 ranges of Protea eximia and Protea Suzanni which do not overlap. However, at 1 and 2 they do overlap. This is because Protea Suzanni was introduced to this region through a process of long-range dispersal, and they were transported here. Specimens were taken from 3, 4, and 5 to look for SNPs to identify the species. The Stadens Wildflower Reserve lies outside of PE. In 1984, the Suzanni were introduced into the reserve and grown and cultivated in the reserve by the then reserve manager. From there, they spread, and it was our idea that they had hybridized with the natural Protea eximia species that exist on Lady Slipper and the surrounding Van Stadens Wildflower Reserve. This is the eximia pictured here and the Suzanni pictured there. For the genetic analysis, DNA extraction took place where leaf material was extracted to produce DNA material. Sanger sequencing was done on the internal transcript spacer from the nuclear ribosome cistron, and we used the sequences to detect hybrids. One site was used to identify the hybrids. This was site 234 of the internal transcript spacer 1. G was for guanine, which was identified to be the Protea eximia. T was for thymine, which was used to identify Protea Suzanni at that site. And K was the hybrid of both, which was identified as the hybrid site. This was according to the TWISPs idea by Potts et al. 2014, or inter-individual site polymorphisms, which show that you have your guanine, your thymine, and then your K, which is a mixture of both, which was shown to identify the hybrids, which are highlighted here in orange. The morphology of the plants, you can see, is very similar between the hybrids and the pure eximia and the hybrids and the pure Suzanni. We use morphological results to look at whether the leaf length and the leaf width would have any effect on how the hybrids were found. And we found that the Protea eximia plants had very broad and short leaves, and the protea Suzanne had very long, narrow leaves, and then the hybrids had characteristics of both. We used HRM analysis because it was much cheaper than using Sanger sequencing. On our initial sample set, we got very good results with high levels of accuracy. But when we started using more samples from a broader range of populations, the accuracy dropped below levels that we required, and hybrid detection became very inaccurate. All the way up to finding single nucleotide polymorphisms, we were sorted. We had identified a specific way of identifying hybrid proteas. However, the HRM analysis did not work out properly, and it requires further work to be able to detect hybrids from the field between these two species. This whole hybrid detection process was just for these two species and couldn't be replicated for other species within the Proteaceae family. Some terminology relevant to my study is non-demonic intrusion, which is defined by Hilbert 1984 as the impingement of chance events on an experiment in progress. And this was its fire that occurred in the reserve in June 2017. The fire was one of the hottest fires on record in years in the reserve, and it took out all my samples that I wanted to actually study. So I was left with the 24 samples I had left to make a map to show the distribution of the hybrids as regards to the Protea eximia and Protea Suzanne. And this was the original area where the Suzanne were planted. Our initial plan was to use HRM. We found that it didn't work, so we used Seedlings and a primer detection method which has been used successfully to identify various species of sharks from thin material. We tried this as a second cheap method. For the species-specific primers, we used a 420 base pair difference across ITS1 and a 234 base pair difference across ITS2. We used A to differentiate between Protea eximia and C to differentiate Protea Suzanne. Species-specific primers picked up 18 hybrids that Sanger sequencing suggested were Protea eximia. I can suggest from this that the peaks of Sanger sequencing were not significantly detectable. This could infer a third or second generation hybrid due to SSP sensitivity. There was a minor decrease in the frequency of hybrids identified via Sanger sequencing from the pre-fire plants to the post-fire seedlings. This was due to 18 samples that were pre-eximia according to Sanger sequencing, but hybrids according to the species-specific primers. There was an increase in the frequency of hybrids to pure species from the pre-fire plants to the post-fire seedlings using the species-specific primers. These results were shown to be statistically significant due to the chi-square test results. This map shows the distribution of pre-fire plants across the reserve. Hybrids had been identified at each of the different sites. This map shows the proliferation of hybrids in the post-fire seedlings. They were mainly in 3 small clumps, here, here, and here, but there were also hybrids here. So in summary, this is the first publication of hybridization due to long-distance dispersal in the proteas. It maps out a way of doing it for similar species. Hybridization in Frankenflora is something we all need to be aware of. After several generations, the original Protea Suzanne species population at Stadens Wildflower Reserve appears to have disappeared, but it has been polluted into the Protea eximia population, as my study has shown. We are uncertain of how far this pollution rate goes. A future recommendation for this study would be to look at the witch's broom, which is shown here. The witch's broom yield appears to be higher in samples that are of hybrid than those that are of pure Eximia or pure Suzanne samples, and these are suggested by field observations. But it still needs to be further studied. This could be because of the lower fitness of these individuals, potentially a negative consequence of their hybridization. Moving lots of species in the Cape, which is known for its highly localized species distributions, is a problem. There could be many hybridization events going on which we are unaware of within this rich genera. We need to investigate this in future using a similar approach to what myself and my colleagues have done to see whether hybridization does threaten biodiversity in range-restricted species. I'd like to thank all the following people for their help on my project. I'd like to thank the following for funding my project. And I'd like to thank all of you. Any questions?